Normally, fertilization takes place, of course, inside the woman's body, where the egg cell and the sperm cell come together, and then when the sperm cell fertilizes the egg cell to form a zygote. However, in the 1970s, the technique called in vitro fertilization was developed, and here the egg cell and the sperm cell fertilizes the egg cell outside of the human body inside a Petri dish, and this process is called, also often abbreviated as IVF, and is nowadays a very common and standard procedure for couples who have problems becoming pregnant the natural way. So I'm going to first explain you the diagram over here, and then over here on the right side I'm going to outline the steps a little bit further. There are some preparatory steps also necessary which are not explained over here in the diagram, but I'll simply explain the diagram first. So, over here, the eggs are collected after ovulation or directly from the follicles, and the sperm are collected as well. So what we have over here is, this is the ovary, and inside the ovary we have a follicle with the ovum, the egg cell, or also known as the oocyte. And this egg cell is extracted using a catheter. So this is a procedure in which the needle is inserted through the wall of the vagina. On the outside there is an ultrasound device which is important for navigating the device. So it is quite a high-tech procedure. Normally the doctors do not only collect one egg cell but multiple egg cells. And in order to obtain multiple egg cells, you need to do a hormone treatment, which I'm going to explain over here later. The sperm cells are also collected, and they have to be concentrated and cleaned, because sometimes the couples are infertile because of a problem of the man, because the sperm quality is too low, or the sperm concentration can be too low. And therefore, it's necessary to prepare also the sperm cells. And then you have several egg cells in your Petri dish. You add them over here, and then you add the sperm cells over here, and then you basically incubate them overnight. And the egg and the sperm cells will find each other, and then hopefully fertilization is going to happen. And that's basically already it. But what I'm going to do now is we have to backtrack a little bit, because what they do not show over here on the picture is the hormone treatment that the woman has to undergo before IVF. And that's what I'm going to explain now. First of all, the woman has to take a drug. Usually it's some kind of a nasal spray. And there are substances in there that stop the production of the hormones FSH and LH. So this means the menstrual cycle is stopped by taking in certain hormones which block the formation of FSH, LH. So I wrote down here down regulation. It would be probably better to, or easier to say, stop the menstrual cycle. So we first stop it, because this gives the doctor more control over timing the whole procedure. We move on, and then the woman is being injected large doses of FSH and LH. So probably much more than she would normally produce herself. And because of this injection of large FSH levels, many follicles start to develop, not only one. Normally, only one follicle develops per month, but if you overdose, so to say, if you inject a lot of FSH, then many follicles are going to develop. So we cause the woman to so-called to superovulate. Now, there is another hormone called HCG, human choriogonadotropin. This hormone is responsible for maturing the egg cell inside the follicle. This is for maturation and loosening of the egg cell. So that's basically, first we stop it, then we cause a superovulation, and then we can insert the device to extract the egg cells. And that's basically where we're already here in the diagram. So steps 1 and 2 are not shown on the diagram, so I kind of had to backtrack a little bit. So you retrieve the egg. Yeah, that's also something that I already mentioned. Fertilization happens because the eggs are mixed with the sperm, and then you incubate them at a constant 37 degrees overnight, and the sperm cells and egg cells are going to find each other, and fertilization is going to happen. And then what you have is, if you have multiple egg cells in your petri dish, then you of course also have multiple embryos being formed, and then you choose, the doctor selects the embryos and implants them, and I'm going to explain this over here again. The fertilized zygote, the fertilized egg cell is a zygote, well, is then removed from the petri dish for implantation. So you take it up again, and then you see it over here, it's sucked in over here, and then it is injected into the uterus wall. It says over here it's surgically implanted into the endometrium. The endometrium is the inside lining over here. And after it's implanted, hopefully pregnancy is going to develop. And additionally, what happens is that the doctor injects progesterone. Remember that progesterone is the hormone which maintains the uterus lining. So it kind of supports and assists pregnancy. And after pregnancy has been established, regular pregnancy checks can be done, and the pregnancy is just as normal. Now, there are certain ethical concerns. And one ethical concern that people have discussed is so-called pre-implantation diagnostics. And there is another ethical concern that I'd like to mention, and that's the implantation of multiple embryos. So maybe I should have written this the other way around. The ethical concern should maybe be the heading over here. But pre-implantation diagnostics is an issue, and also the implantation of multiple embryos. Multiple embryos implanted. So why are these ethical concerns? There are in certain countries laws on how many embryos you are allowed to implant at the same time. So in some countries the maximum is 3 embryos, with the hope that one of them is going to develop. They do that, they have these laws because you do not want to implant, let's say, 5 or 6 embryos, and then later on you have to abort some of them to have a safe pregnancy. So that is the reason why there is a certain maximum number of embryos that can be implanted at one time. And this is often regulated by the national law. And another issue, which is ethical concern or ethical issue, is the possibility of so-called pre-implantation diagnostics. So, and this means is, theoretically, if you have your 10 embryos over here in your petri dish, you can do a DNA test on them. Because you can remove one single cell, they already started to divide, and you can take one cell, do a DNA test, and then you can choose which embryo you want to implant. So you can essentially test for gender, you can also test for the presence of certain genetic conditions maybe, and then it's possible to maybe fulfill the wish of their parents for a certain gender of the baby. Now, you might wonder, well, that's actually quite nice, so what's so problematic about this? Why is this an ethical concern? The thing is the following, that essentially by doing that, we're not just modifying or changing one generation, but essentially all following generations. So if there's a social pressure to have boys, for example, and this pressure does exist in many cultures and countries, then of course you're going to have parents select boys to be implanted, and this might cause or will cause an imbalance between boys and girls also in the future generations. A counter-argument to that would be, of course, that, well, right now you already have abortions of girls, where girls are more likely to be aborted than boys. So is it not better to simply select at an earlier stage than to have a later abortion? Now that would be a counter-argument to my previous claim. So basically what I would like to say is that all of these modern techniques, while they have many advantages, also give rise to a range of ethical issues. I can add a third one if you want to. Over here is that the whole hormone treatment that the woman has to undergo, there are certain health risks involved. There are health risks. There are higher risks because of the high levels of FSH and LH and the high levels of hormones being injected. This can increase the risk of certain cancers in the woman. So you have to balance now the wish for a child with the associated health risks. So the list goes on. The list goes on. And what I probably would do is I would probably cancel this out here and write it over here, ethical issues. And that's, of course, quite offers quite a large basis for discussion. And if you want to do your own research, it's kind of interesting to see how different cultures and societies weigh these different issues. Certain issues are not considered a big issue by some countries and some legal contexts. Others are considered to be very, very important. So that's also kind of interesting for me, at least, to see how different societies and cultures and also legal systems deal with the issue of IVF differently. Just to add as a last interesting comment, just to show how far this goes, instead of simply killing the remaining embryos which are not needed because they were not implanted, some couples in some countries it's allowed to give them up for adoption. So what you do is you basically take an embryo from another couple and implant that, so the woman becomes pregnant with basically with her own child, but it's not the genetic child. It's the DNA of another man and woman in the embryo. So that's also possible, and all these things have to be regulated by law and have to be regulated by certain social norms. Okay, I think that's the last thing I just want to say. Again, over here, a very good practice would be is to summarize the whole IVF process in a nice paragraph.